All right, guys, today we're going to talk about cell respiration. And we've kind of mentioned this a couple times before, but cell respiration, why do we do cell respiration? It's because all cells need energy, right? And we know we need that energy to do any type of life process, right? Like if we're moving muscles, if we're digesting something, and remember that's an every organism kind of thing. And so where does cell respiration take place? This is going to be in our mitochondria, right? And remember, mitochondria, this is an organelle. And where do we find organelles? Organelles, we're going to find them in eukaryote cells. And who are my eukaryotes? My eukaryotes are my plants, my animals, and my fungi, okay? So all eukaryote cells have a mitochondria. All eukaryote cells go through this process. So since this is going to be a reaction, we need to talk about what we need, okay? So what I need for cell respiration is I need glucose, and I'm going to need oxygen. We'll talk about why we need my oxygen here in just a little bit. All right, and then what the purpose is, right? Like, what am I making in this reaction? I'm making ATP, which is our big take-home thing because ATP is energy, but I'm also going to make some carbon dioxide, and I'm going to make water. Because these are things I'm making, but I don't actually need, this is called a byproduct, okay? So this over here, my ATP, this is the product I'm trying to make. This is like my end goal. These two things over here, the CO2 and the H2O, these are things that I'm making as a result, but I don't actually need. All right, so then when I'm looking at this, since I need oxygen, we actually get to call this a special type of respiration. This is called aerobic respiration. Because when we do aerobics, think about like exercising and stuff, what do you need a lot of? You need a lot of oxygen, and that's what aerobic is. And what do we know about oxygen? Well, we know oxygen is super negative, right? And because he's super negative, that means he is, we talked about before, he's really greedy, and he likes to take electrons. So he likes to take electrons. And that's going to be really important to make this whole process work, actually. So, since this is a chemical reaction, guess what? We get to write a chemical equation. So here is my equation for aerobic cell respiration. I need glucose, I need oxygen, and that is going to make 36, now we get to have a number with this. This is going to make 36 ATP, right? This is what I want. And then I'm also going to make the water. I'm also going to make CO2. Now remember, these two things right here, these are my byproducts, something that's being made, but I don't necessarily need. The 36 ATP, this is what I want. And then, of course, since it's a chemical reaction, I can name the things I have to do it. These are called my reactants, okay? Now, as far as aerobic respiration goes, I actually get to do this in 3 stages, okay? So I have 3 stages. My first stage, this is called glycolysis. My second stage is called Krebs cycle, or it's called my citric acid cycle. And then my third stage, this is called the electron transport chain. All right, so let's go through what's happening in each of these stages, okay? So my first stage, stage 1, This is glycolysis. So let's talk about glycolysis. Let's talk about what this word means. Glyc, we know what glyc means. Glyc, let's get rid of that one. Glyc is referring to sugar. And what is the sugar we're using? The sugar we're using is glucose. And then what's lysis mean? So we've done this a bunch of times. Lysis means to break. So what am I doing during glycolysis? I am breaking apart my sugar, right? All right, location-wise, this is going to happen in my cytosol. All right, now remember the cytosol, that's all the empty space within my cell. And so since this happens in the cytosol, this glycolysis, this is going to be a universal reaction. And what I mean by universal reaction is that every living organism is capable of doing glycolysis because every organism has cytosol. All right, so let's follow what's happening in glycolysis. All right, first we're going to start with our glucose. Now remember, glucose formula is C6H12O6. But since we're doing something with a living system, what element out of these three am I going to focus on? Since I'm doing it, I'm organic. I'm going to focus on the carbons. So I'm just going to make this a little bit more simplistic. I'm going to take, because see it's C6, so I did 6 C's. 1, 2, 3, 4, 5, 6. I have 6 C's right here, so that's going to be my glucose. All right, now you guys tell me, what do I do? Here, I'll go ahead and label it. This is glucose. What do I do, or what do I have to do whenever I want to get energy out of a molecule? I have to break it. And I have 6 C's right here. So what's going to be the natural way I can break this? I can break this into 2. So I have 3 carbons over here, and I have 3 carbons over here. When I break a chemical bond, that releases energy. The energy, so I can get 2 ATPs here, and then I also get to make 2 NADHs. So we need to talk about him here in a second, my 2 NADHs. But if I go all the way back up to my equation, I'm supposed to get 36 ATP. Right now, I have 2. So that means that's why we have to go through the other steps in aerobic respiration, is because I need to continue to make more ATP. But that's a good start right there. Now these 2 molecules, these 2 3-carbon molecules, they each have a name. These are called pyruvate, or they can be called pyruvic acid, okay? So let's talk about NADH really quickly before we move on. NADH, he's going to be important to us later because NADHs, these are really, really good electron donors. All right, say they give up their electrons really, really easily. And since they give up those electrons really easily, I can break them apart to get those electrons, and then I can use those electrons in another part of my reaction. Okay, so just remember that. NADH are really good electron donors, and I'm going to need them later on. Okay, so that's glycolysis. That's my first step. Or there's only 3, technically. There's only 3. And the first one is glycolysis, where I take sugar, I break it apart, I get energy. But it's not enough energy to fuel my life processes, so I have to keep going on. Now, we didn't list this one because it's like step 1.5, okay? Now, my step 1.5, this is my transition phase. And whenever you transition, it means you're going from one place to the other. And so all of these things, like these 2 pyruvates, I'm going to move them from the cytosol, and I'm going to shove them all the way into the matrix. Now remember, if this is my mitochondria, right, and then here's the squiggles, the matrix is this inside part. So not the cristae, but the part inside the cristae. And so this transition phase, that's what I'm doing, is I'm taking these pyruvates and I'm going to shove them all the way into the matrix. But here's the deal. So here's where I start. Here's my 2 pyruvates, okay? Remember, these are pyruvate or pyruvic acid. Whenever I have to shove them through that mitochondrial membrane, right? And I have to shove them through the cristae, right? So I'm going from that cytosol to the matrix. So whenever I shove them into that mitochondria, that means they break apart a little bit and I lose a carbon. And so now, when I shove them through that membrane, okay, so this right here, this is when they go through the membrane, they lose a carbon. All right, now what do they lose that carbon as? That's actually lost as CO2, right? That was one of our byproducts. I don't need him, but he was made regardless. And so every time you guys breathe out, just as you're breathing, sitting there, every time you breathe out, that CO2 is because you're taking stuff from your cell and you're shoving it from the cytosol to go inside your mitochondria. And that's why you breathe out CO2 every day, since the day you were born until the day you die. When you breathe out CO2, it's because you're shoving stuff from the inside of your cell into your mitochondria. Now, here's the fun thing, though. Because I broke those 2 pyruvates, remember I lost a carbon, that means I get to make a little bit of energy. I get to make, well, not energy. I get to make my good old friend NADH. So I made another 2 NADHs by doing that. So far, up here in glycolysis, I've made 2 ATPs. I've made 2 NADHs here. In this transition phase, I got to make another 2 NADHs because I broke my carbons. All right, well, guess what? Because he is a different molecule now, right? I'm not pyruvate because I lost a carbon. I get to have a new name. My new name is acetyl-CoA, right? And then this guy's name is acetyl-CoA. And that's what I get to start with in my next phase, which is, let's use this color. So this is stage 2. This is going to be my Krebs cycle, named after, guess who? Hans Krebs, because he's the one that figured this out. But it's also called the citric acid cycle, because that's one of the molecules that involved. So you guys know how citruses like oranges and lemons and limes have that taste? Well, you guys have citric acid inside your cells too. What do you know? So what we're going to do is we're going to start with our molecules. So I'm going to start with these 2 acetyl-CoAs. So here's 2 acetyl, here's another acetyl. So these right here, these are my acetyl coenzyme A. And what I'm going to do is I'm going to break these suckers apart, okay? I'm going to break them apart. And what do I get? Every time I break chemical bonds apart, I start getting energy. And so when I break these apart, I actually get 3 things. I'm going to get 3 NADHs, remember, good electron donors, so I get 3 from each one. So 3 NADHs, 3 NADHs. When I break this acetyl-CoA apart, I get to have another new friend who is an FADH. And FADH is the same as NADH. He's really good electron donors. They like to give them away. And they also get to make 1 ATP for every acetyl-CoA molecule. So I just took that acetyl-CoA and then I crushed it up as much as I could, and all I have left now is energy and then really good electron donors, which again is going to be important here in this last step. So far, I had my sugar, right? This is where I started. I had my sugar. I broke it apart to make pyruvate. I got energy because of it. Then I had to shove him into the matrix. Because I shoved him into the matrix, I went from pyruvate to acetyl-CoAs, and I got a little bit of energy. And now I'm in the Krebs cycle, which is, let's go ahead and write this out to the side. Krebs cycle, this is going to be in the matrix of the mitochondria. And I took those acetyl-CoAs and I broke them down into either energy donors or I broke them into ATP. But I need to finish this whole reaction because I need to finish making the rest of my ATPs, and I get to do my last reaction. Well, there's only 3. So my last reaction, this is the electron transport chain. Okay, electron transport chain. Now, the electron transport chain, we talked about this last time. This is happening on the cristae. You remember cristae is my awesome little bilayer, and he has ATP synthase in him. All right, so this is where we're going to make that ATP synthase spin. All right, so coming into the electron transport chain, we can kind of do a tally, right? Well, okay, let's do it this way. So let's draw my cristae. So here's my phospholipid bilayer, and then here's my ATP synthase. Remember, ATP synthase is the enzyme that makes ATP. So here it is. This is my cristae, right there. And this guy, let's go ahead and label it. This guy right here, this is my ATP synthase. All right, so what happens is I've made all of these electron donors, right? So I've made these NADHs and I've made these FADHs. Remember, both of these are really good electron donors. They like to give their electrons away. So what's going to happen is I'm going to break both of them apart, which is going to release electrons. And you know what those electrons do? They're going to make the ATP synthase spin. ATP synthase spin. Because remember, if right here, like these are my 2 active sites, yes? And then I have my 2 substrates. I have ADP, sorry, that's a P. And then I have my phosphate. And so when they go sit inside that active site, and I want to join them together, this wheel up at the top has to spin. Well, to make him spin, that's where I need the electrons from the NADH and the FADH, right? So these electrons help the wheel spin. And when that wheel spins, that's going to take the ADP and the PN, it's going to force them together, and then out the other side, I'm going to get, let's make it a nice color. This is where I'm going to get my, oops, that's not an A. This is where I'm going to get my ATP molecule. Okay, so that's why for those couple reactions, I kept making these NADHs and these FADHs, because they will give me the electrons. Those electrons will make that top part spin, and when that spin, that forces the ADP and the P together, and that will give me ATP. Now, as a note, and it sounds kind of not important, but I promise it is, we also have to have oxygen to do this. So we need oxygen, because that oxygen, he is going to help accept the electrons, is really what he does, okay? So if I didn't have oxygen here, I wouldn't be able to get the electrons out of those NADHs and those FADHs, and if I don't get the electrons out of here, then my ATP synthase doesn't get to spin. If he doesn't spin, then I don't get to make ATP. So oxygen, it sounds kind of like a side note, but he's actually really important to the overall scheme of things. And because oxygen's sitting out here, and then when I break apart that NADH and that FADH, I also have some leftover hydrogens. And whenever oxygen and hydrogens come together, guess what? I get to make water. So remember, water, this was another This was another byproduct, right? So every time you guys breathe out and you go, and you breathe out the CO2, remember you're also breathing out some water. Well, that water is coming from this electron transport chain because the oxygen was sitting there taking electrons from the NADH and the FADH so we can make the ATP synthase spin so I can make my ATP. So let's get to our final numbers, right? When we came into, so coming into, I'm going to put the ETC, the electron transport chain, I had 10 NADHs, right? But if you recall, let's go all the way back up here. Glycolysis, let's do it in red. Glycolysis, I had 2 right here. Then during transition phase, I made another 2. So 2 plus 2 is 4. And then during the citric acid cycle, I got to make another 6. So I had 4, 5, 6, 7, 8, 9, 10. So that's why down here, coming to the electron transport chain, I had 10 NADHs. Now my FADHs, I had 2 FADHs because I only made them right here at the Krebs cycle or citric acid. I made 1 right here, I made 1 right here, so I had 2. Now we get to use an awesome little conversion. For every 1 NADH, that allows the ATP synthase to spin and make 3 ATPs. Well, if I had 10 NADHs, so 10 times 3, that gives me 30 ATPs. Remember, my final number was 36, so right now I have 30. For FADH, for every 1 FADH, I get to make 2 ATPs. Well, I had 2 coming in, so 2 times 2, that'll give me 4 ATPs. So overall, right now, I've made 30 from NADHs. I've made 4 from my FADHs. So that gives me a 34 ATP. I'm trying to get to 36. And so if I go back up to glycolysis, oh yes, right here, look, I made 2 ATPs. And then I also made 2 right here. 2 right here in Krebs. So 2 plus 2 is 4. There's another 4 ATPs that I made throughout all the reactions. Now I'm at 38 ATP, which is more than what I wanted. But as with anything, when I crossed, so when I cross into the mitochondria, that cost me 2 ATPs. So 38 minus 2 will give me that rousing number of 36. Okay, so let's recap really quickly. Let's recap in this lovely electric blue color. So cell respiration. I do cell respiration because I need energy. All eukaryote cells, all living things need energy, but all eukaryote cells are going to do this in the mitochondria because, as you guys love to say, it's a powerhouse, and that's where I make the most. So to do this reaction, I need oxygen and glucose because I need glucose to break it apart so I can start making all my molecules. I need oxygen because he's really greedy and he's going to take the electrons from those NADHs and those FADHs. I make CO2 and H2O as byproducts. I don't care about them, but they are made regardless. This is aerobic respiration because oxygen is involved. And again, I'm trying to make the 36 ATP. I have my 3 steps: glycolysis, citric acid cycle, electron transport chain. Glycolysis breaks my sugar apart. I get ATP, and then I get some good old electron donors. Transition is when I go from out here in the cytosol all the way inside my matrix. When I do, it breaks it apart a little bit more. I get another electron donor. Citric acid cycle takes my final molecule, breaks that sucker apart. I get a bunch of electron donors, and then I get a little bit more ATP. And then once I get to my last step for the electron transport chain, my entire goal is to make this top part of ATP synthase spin I need my NADHs and my FADHs to give those electrons so he can spin. And then when he spins, that means I get my final thing, which is what I want right here, is that ATP so I can fuel all my life processes. Oxygen is there because he's going to help take the electrons from the NADH and the FADH. I get water as a byproduct because I'll have some oxygen and some hydrogen floating around. And then from all the conversions and subtracting a couple of things, I get the 36 ATP. Okay, I hope that helps.